Assalamualaikum warahmatullahi wabarakatuh and a very good day to everyone. Today we are from group 2 will be presenting about chemiosmosis in photosynthesis, which is the core of all photosynthesis process in all plants. But before that, I am going to introduce to you Peter Mitchell, a Nobel Prize winner. He won this award by his discovery of the chemiosmotic mechanism of ATP synthesis. So this hypothesis stated that a proton motive force was used to drive the synthesis of ATP. So now let's get down to real business. What is chemiosmosis? It is a term that is used to describe the movements of ions by diffusion across a semi-permeable membrane, going down the concentration gradient. In plants, chemiosmosis occurs in chloroplasts, where ion diffuses through the thylakoid membrane. Now, let's move on to the next part.
Okay, so I'm going to proceed to the mechanism of chemiosmosis. Firstly, when the light energy is captured by the photosystem II, the electrons within photosystem II get excited and causes the water molecules to undergo photolysis. The photolysis of water molecules breaks down the molecules into protons, oxygen molecules, and electrons. The protons is transferred into the thylakoid space, whereas the electrons will be captured by the plastoquinone. However, oxygen molecules will be transferred out from the organelles. Next, this is showing how the light energy is captured by the photosystem too. Next, the electrons captured by the plastoquinone will be transferred into the cytochrome complex. This complex also carries the protons from the stroma space into the thylakoid space. This event further increases the concentration of protons, which is higher in the thylakoid space compared to the stroma. This imbalance of concentration of protons will lead to the increasing of pH value, which obviously will disrupt the homeostasis, which is why these protons need to be transferred out from the thylakoid space by the help of ATP synthase. The movement of protons into the ATP synthase provides energy for the phosphate to be added to the ADP, which then converted into ATP form of energy. This form of energy will be used into the next cycle, which is the Calvin cycle. So that's all from me. Thank you.
So for the next point of chemiosmosis is the importance of it. The first thing first is that we need to remember that the purpose of light-dependent reaction is to produce ATP and NADPH that will be used in the next process. As you can see from the first illustration here, the ATP here will be used in the first stage of Calvin cycle, which is known as the carbon fixation stage, whereby 6 ATP will be converted into 6 ADP. Now, the question of the day is, what happens if ATP is unable to be produced? And the answer for this is pretty straightforward, which is that the dark reaction cannot occur, thus the glucose cannot be produced at the end of the photosynthesis. In a nutshell, this means that the photosynthesis process is halted in the middle. So this brings the significance of the chemiosmosis process, which is that chemiosmosis produces ATP molecules for the continuity of the process of photosynthesis.
So now let's move on to the last part, which is the differences and similarities of chemiosmosis in mitochondria and chloroplasts. There are two similarities of chemiosmosis in mitochondria and chloroplasts. The first one is both of them involve electron transport chain. The second is both of them use ATP synthase as their carrier proteins to transport the protons across the membrane. Next is the differences of chemiosmosis in mitochondria and chloroplasts. which can be categorized in 2 points: special organization and ATP. The first difference is in the mitochondria, the chemiosmosis begins inside the mitochondrial matrix, while in chloroplast, it begins in stroma. The second difference is about ATP synthesis, where in mitochondria, it embedded inside the cristae. For chloroplast, it embedded inside the thylakoid membrane. And for the last one is about ATP. The ATP produced in mitochondria are the product of glucose breakdown, while in chloroplast it is used to build glucose.
In conclusion, concentration gradient is a key component of the biological world. As we have presented before, chloroplasts generate the ATP by a mechanism called chemiosmosis. In this system, concentration of a molecule in one compartment is higher than the neighboring compartment. A proton motive force is built when the ATP synthase pump a proton across the membrane in form of H+ gradient. The discovery of this chemiosmotic mechanism has been a major success towards the study of plants in today's world.